students. In the previous class, we have studied about the flagella, structure and the number and arrangement of flagella in the bacterial body. Today, we are going to learn about the bacterial gram staining. Okay, bacterial gram staining. Basing upon the cellular compositions, bacteria are divided into two types. They are, basing on the cellular composition, bacteria are divided into two types. They are gram positive and gram negative. So, what is the gram in these bacteria? That is a scientist's name who divided the bacteria into two groups. They are gram positive and gram negative bacteria. So he is Sir Hans Christian Gram. Sir Hans Christian Gram. He is the scientist who performed the gram staining procedure. Now, let's see what is gram positive and gram negative in the structures of these bacteria. So, these are gram positive and these are gram negative ones. So, cellular structure. This is the cell wall and this is the cell membrane. This is cell membrane. And this is the cell wall. You can see the cell wall is thicker and broader. So this is cell wall and this is cell membrane or plasma membrane. So what is the composition of the cell wall in gram positive bacteria is the peptidoglycan and the presence of teichoic acids in the peptidoglycan layer. The presence of teichoic acid. This is the cellular wall composition of the gram positive bacteria. And what about the gram negative ones? Here the gram negative bacteria are made up of 3 layers. This is the outer layer. It is made up of lipoproteins or lipopolysaccharides or lipopolysaccharides. Lipoproteins and polysaccharides. And then the second one, this is cell wall. And the third one is, this is plasma membrane. Third one is plasma membrane. Here there are 3 layers in the gram negative bacteria. So what's the difference between these two is, in gram positive bacteria, peptidoglycan layer is somewhat thicker in size, and in gram negative bacteria the peptidoglycan layer is thinner and there are no teichoic acids in the cell wall. No teichoic acids in the cell wall. And this makes the difference between gram positive and gram negative. So now have a look at the gram staining procedure. So now gram staining procedure. First of all, take a loop full of bacteria. Loop full of bacteria on the slide, and then heat fix it on a Bunsen burner. On Bunsen burner for 30 seconds to 1 minute. 30 seconds to 1 minute, and then add crystal violet dye. Crystal violet dye to it and let it dry for 30 seconds to 1 minute. Next, wash it gently. Wash it gently, and then now add grams iodine. Grams iodine, and let it be for 30 seconds to 1 minute, and then wash gently under the tap water. Under tap water, and then now it is a key point in this procedure. Now all the cells which are gram positive and gram negative, we are staining the bacteria. We don't know what are the cells present in the stain. In the slide, on the slide. So now before adding the acetone or alcohol, we have to look at it, that we have to observe that the gram positive bacteria which have a thicker cell wall with teichoic acids and peptidoglycan layer, it retains the crystal violet dye, and after the wash with acetone or alcohol, the gram negative bacteria loses the dye. Now add the alcohol. Add the alcohol, which is 90% acetone. Now let it be for 30 seconds to 1 minute. And now wash it gently. Now if you observe in a microscope, you can see the cells with crystal violet dye and cells without any dye. So the cells which dyed in violet color, they are gram positive ones, and the cells without any color are gram negative ones. So if you want to color the gram negative ones, so let it color with the counter stain, that is saffronin, which is pink in color. Okay, now the gram negative bacteria also are having the dye which is pink color. If you observe in the microscope, the gram positive bacteria appears crystal violet in color or violet in color, and gram negative bacteria appear in pink color. So we can detect the gram positive and gram negative bacteria on this slide under the microscope. Okay, this is about the gram staining procedure and the differences between gram positive and gram negative bacteria. So in the next class we will discuss about the respiration in bacteria.